plant diseases in the greenhouse setting. So whenever we're dealing with plant diseases, we really want to focus on the proactive approach, not the reactive approach. So what I mean by this being proactive, we are going to scout for different signs and symptoms of the plant diseases, maybe seeing their spores, maybe seeing necrotic spots, instead of waiting for the plant to completely decline. And then when the plant declines, we're going to be losing out on money. So we don't want to wait that long. So we want to have the pre-active approach there. And that's how we're going to be getting into scouting of these plant diseases. So like I was saying before, with scouting for plant diseases, disease control really relies on that prevention, preventing that disease from occurring, because the diseases are always present there. It just depends on the climate and the environment for that disease to really inoculate the plant material and create issues. So, and then the second point, by the time you see these symptoms, it's often too late to manage those diseases in the plant. Because that plant is so small, it's going to affect the plant a lot quicker. And when that disease does affect the plant, the decline starts to occur. And yes, you can put different fungicides on there, but it's already past the point that you're going to be able to fix that plant. So really, that prevention approach, even if we are going to spray the fungicides, that we do it preventatively, not reactively. Diseases can come from a lot of different things, really anything that you're dealing with. Infected plant liners that are coming in, seeds, plugs, divisions from different nurseries or greenhouses that you bring in plant material from. Also, residual plants from previous crop or previous seasons, and really that means from your previous season, if you did have some kind of disease that came up, maybe daylily rust that has came up, and that you did not completely sanitize the area and remove all of those plants that were inoculated with the daylily rust. So when that occurs, if you don't get rid of absolutely everything, even just one little spore can inoculate the new plant material for the next season. So that's where that residue of plants from the previous crops or previous seasons comes into play. And then also plant and soil residues and debris from the current year. So that's cleaning up, making sure that everything is sanitized, making sure that there's no weeds that are present, or if you did have any diseases that have came on your plants for that current season, that you get rid of absolutely everything and that you sanitize everything that is done afterwards. That includes all of the tools that we have done in class all throughout the semester. Okay, so here's just an outline of some stuff that we're going to be focusing on throughout this presentations. And I don't want you guys to get overwhelmed or anything like that. I just want to give you a brief overview of different diseases that can come on different greenhouse plants. So the fungus, root rots, leaf spots, rust and mildews, bacteria with the leaf spots, vascular rust, and soft rots. Viruses, nematodes that are going to be coming up, but mostly there's foliar nematodes, not the root rot nematodes or anything like that. And then phytoplasmas or aster yellows. And for the individuals that haven't had like integrated pest management or that are new to this program and new to pathogens in general, I just wanted to show you guys the disease triangle. I mentioned earlier that the pathogen is always going to be present. And depending on, the environment will depend on if the inoculation will occur with the host plant. So here's the disease triangle, and you have to have all of these three factors into play come across for the pathogen to get inoculated onto the host plant to be affected by that. So we need the host, right? So in that case, that daylily rust, it would be the daylily. We need the pathogen, which would be the rust, and then we need the correct environment. And usually this varies depending on what kind of disease that you're dealing with, but it's those moist conditions, very high temperatures. It could go from about 72 to 80 depending on where you're at, but you don't want it too high and you also don't want it too low, but we also need that moisture also. So we need all three of those factors, the host, the pathogen, and the environment, to have that inoculation of the pathogen to the host to occur and to the host to start declining from the pathogen. And then here's a really good example. We have all three of them coming together, and you can see in the dead center that we have that disease that has been created. So we have the stressed or injured plant, the host, the pathogen, that disease that's causing it, and then the environment with the wet foliage or soils, high humidity, and then poor air circulation. Okay, so for the disease management principles, we have that first one, eliminate that initial inoculum or the pathogen survival. And we mentioned that earlier with the different previous years of the residuals of the diseases, that we need to get rid of everything. So that's really key with sanitation. With sanitation, we want to remove that initial inoculum, the inoculum being the pathogen itself, and we want to completely remove that as much as possible. I know it's not a good thing to say 100%, but we want to try to remove it as much as possible. So what we could do is that sanitation and then scouting for early detection on that, and that's going out every single week. If you do have your own greenhouse or plant propagating facility, that you go even every single day, have someone that is designated to go out there to check for different diseases that may be arising. Either every day, every other day, every 3 days, depending on what crop you have. Different crops are going to be more susceptible to these diseases. So really determining that, but having someone designated for scouting, because if we get that early detection, if we see, say, powdery mildew that is starting to affect some of the crops, we can go in there and apply the fungicides, try to create more air circulation in there to decrease that disease from spreading throughout the other plants as well. Another one, besides the elimination of that initial inoculum, we can reduce the pathogen spread. And you can do that by reducing water splash. So we were talking about, overhead irrigation, right? When we do overhead irrigation, there's a lot of reasons why it's bad, but another one that I really didn't mention is that water splash. And whenever we're doing the overhead irrigation, that is going to make the plant more prone for water splashing. And water splashing is just when the water falls on the soil surface and it splashes to another plant. Because, with greenhouses, the pots are very close together to maximize space. So water splash can occur at any time, especially with that overhead irrigation. So we're going to want to be very careful that we reduce water splash. And you guys know another alternative for overhead irrigation, one that I'm very fond of, and that's the bottom feeding, which you can do as well. So we can reduce water splash that way. And then we also have that plant-to-plant contact. And with that, putting plants so close together that the neighbor plant is actually touching the other neighbor plant. So we want to make sure that the foliage is not touching each other by just placing the plants further apart. So this is going to depend on what plant species you have, the crop that you're dealing with and how big the plant is. You want to make sure that these are not touching each other. And another one to make sure, yes, it's not touching each other now because there's no, airflow or anything like that, like the fans are not on. Put the fan on, and even when the fan is on, you want to make sure those plants are not touching each other, because with the fan in play, you're going to have plants that are, going a bunch of different ways, not just standing still. And when that's occurring, that plant-to-plant contact might be more prone. So be very careful that you deal with that as well. Wind dispersal, we kind of just mentioned that with the fan in a greenhouse setting, you have more covered area. So wind is not going to play a role or anything like that, but when it comes to the fans, wind dispersal can lead to pathogen spread. So we want to reduce that, and you could even do that by lowering the fans. You still want to have that good air circulation, but even lowering the intensity of the fan will be beneficial. Lastly, we have vectors, and vectors are usually sucking insects that transmit the virus or different diseases or parasites from one plant to another. So we want to be very careful with managing our insects, which we're going to get into in the next chapter of this week with insect management, insect and mite management. But we also are vectors ourselves, with human beings. And we talked about that with a seed dispersal, that we as humans can disperse seeds ourselves by either walking, getting it on our clothing, maybe we would be eating something, and it could also get on our feet as well with the seeds, but that can be the same thing with the inoculum. The inoculum can get on our shoes, it can get on our clothing as well. So us as workers, we need to be very careful that we are not spreading the pathogen either. And lastly, we have tools. And we already got in this too, that we could spread the different inoculum from our tools that we use. So pruners, whenever we're pruning something, we always want to make sure that we're putting it into some kind of alcohol solution or bleach to reduce that inoculum as well. So make sure that as tools that we are not spreading those pathogens that way. Okay, now we just are going to get into different products that are available if we do have different diseases that are emerging, what we can use to prevent these diseases or manage these diseases. So if you guys are not familiar with products, it's okay, it'll be fine. So we could just take it, the basics. But fungicides, primary disease management tool, the fungicides are for fungus or fungi. Bactericides like coppers and phosphonates are for bacteria and cannot be used for a fungicide, and a fungicide cannot be used for a bactericide or bacteria. And then we also have biological control, which is getting a lot more popular. And biological control is using different strains of bacteria. They've used different insects in the past, different things that are natural that are going to be able to use to prevent these diseases or manage these diseases, and the fungicides and bactericides. It usually relies on, the predation from these different organisms or the parasitism from the natural, just like natural mechanisms that have evolved in these different organisms for many years. Like I said, it's becoming a lot more popular, and the efficacy is not where it is with the fungicides and bactericides that are used right now, but I could definitely see the efficacy increasing in years to come. It's just right now, it's not necessarily new because it's been going on for decades, but it's becoming a lot more popular now that the more organics that are becoming popular. So now the biological controls are following, but we really need to make sure that efficacy is still there. But it beats using products and different chemicals all the time, money-wise, and just for the different biologicals, the good organisms that are in the greenhouse as well. So that's a different method other than just using chemicals to decrease these different diseases. So whenever we're scouting, you really want to concentrate on the entryways into the greenhouse, like doors, the open vents, or walkways, because in these areas, a lot of that residual inoculum is going to be occurring. You have people that are coming in from, outside and in different areas that are coming in, and that could pass along the inoculum to the different plant material. So usually those are the first ones to be affected. Another, concentrate on the incoming plants that have just arrived from different greenhouses other than yours, because those, as you saw in the first couple of slides, can be the plants that are going to have the initial inoculum. So we want to make sure that those are not diseased, that we want to make sure that those are disease-free, but also watch those for the first couple of days or first couple of weeks, because even though that we don't see it as soon as it comes in, it's not to say that it hasn't been developing for a couple of days and the symptoms of the disease are not present yet. So you definitely want to just watch those plants and make sure to watch those plants for the first couple of weeks, even going every day as you would be scouting. And then you also want to look out for the out of the ordinary plants, and those are stunted. Stunted is when it's reduced growth, and it could also be off color, usually with a lot of the plants, depending on what you're working with. I don't know if you're working with a variegated plant or not, but if a natural, normal plant is off color, you really want to see if it has that dark green look to it with those healthy, large leaves. So if it's off color, maybe it's yellow, maybe it's light green, maybe purple. We have yellowing, wilting. Wilting is when the plant just looks like it's dry and it looks like it's drooping, and it loses vigor and it becomes very limp. So you would also be looking for wilting, browning of the foliage. Maybe it's distorted. Maybe it just looks different from all the other plants that are growing straight out. Maybe it's curling. And then you also look for leaf spotting, and that's just like different spots on the foliage that aren't normal for that specific plant species. All right, if your greenhouse facility focuses on a lot of like root and cutting, some of these diseases can occur. Some common diseases are the damping off and root rots. And these spread very quickly, so you need to manage these tremendously. So the symptoms include wilting, leaf yellowing, plant stunting, softening, and discoloration of the roots and plant death. You can tell the discoloration by the roots. The roots should be nice and healthy. It's like a cream color. Whenever you have damping off or root rot diseases, you look at the root system and they're brown. They look black and they just look dead. And sometimes the areas are healthy, so they can be that cream color, and then you'll see that distortion come into play, and you can see that necrosis come in, so it's black. So it's the cream color, very healthy, and then it goes brown, and then possibly cream color again, or it's all going to be black. So it just depends on what you're dealing with and the severity of the root rot and how far it's came. But that the roots are definitely an indicator, and then the plant itself. But you can tell with the root system, it will show symptoms of root rot within the root system before the plant is wilting. So that's a really good indicator. You could just check the roots and see what kind of roots that you're dealing with, to see if they're nice and healthy, or if you are seeing those black spots or not. And those major pathogens are Pythium, Phytophthora, root rot, and then Rhizoctonia that you're going to be dealing with. And these diseases favor very wet rooting medium, long-term moisture. So you don't want to have that moisture continuously be wet. We need a little bit of dryness, but we don't want the whole entire root system to dry out. So we really want to find that happy medium. High humidity, poor ventilation. And the poor ventilation usually is occurred if you look on the left of that picture. You see a lot of those plants that are coming together that are so close, so that foliage is touching each other. There's not so much air circulation that is happening here. So that's that poor ventilation. But you could manage this by having, spaces in between your plant material, and you can also have different fans coming in. And high soluble salts, EC, can also play tool in this. So just visually, I wanted to go back to the left picture just quickly, but visually you can see this occurring. If you look at your plants and just a big spot of your plants are missing, so they either completely died or they're declining. So it really starts from that center and then it radiates out. It keeps on, that epicenters in the center, and then it radiates out and radiates out. And by the time, if you don't manage this, all of your plants will die and you have that donut effect. You're going to see like a big circle that keeps on expanding. So that's a good indicator that you might have damping off or root rots in your greenhouse. And then here's just some photos of the root disease symptoms. So here on the top left is that Phytophthora root rot, and you can see the discoloration within the leaves. We have some leaf drop, we have necrotic spots, even a little bit of the leaves are turning purplish. And then we also see wilting on this. To the right, we have Pythium on stonecrop. And we have that discoloration again. You can see that middle stem, how it's like purplish blackish, so we have that discoloration, we have the yellowing leaves, and then the flower is also discolored and almost going a little bit necrotic. And necrotic is just black, it's dead. Necrotic means dead. And then you can see some photos of Rhizoctonia in the bottom two photos, and you can see the wilting and also the chlorosis. FYI, chlorosis means yellowing of the plant, if anyone else needs to know that. And you can also see in the right photo, we have the chlorosis and the dieback occurring. Now we have the black root rot, and the disease favored by this has that high potting medium moisture, and then it has high pH and a lot of fertilization. And you're going to see this come up in a lot of the diseases that with the high amount of fertilization, especially with nitrogen, that these diseases come on. And that's because with a lot of fertilization, with a lot of nitrogen, that means the plant is going to have a big burst of growth. And with a big burst of growth, that plant is more stressed out because it didn't take the time to properly, grow at its own pace, just had a growth spurt. So it's going to be very stressed out. And with stressed plants comes the disease. So that's where that comes in. The spread, water movement, and contaminated pots, trees, and rooting medium. So that's going to be very important with making sure that your pots are sanitized and your trays are sanitized, as well as your rooting medium. And then the control is going to be for just sanitization and fungicides. Botrytis blight or grade mold is going to be a huge issue, and it's always a big issue in greenhouses and managing this. It comes on many different plants, has a wide range of hosts, and the biggest issue is the plant could survive in below 39 degrees Fahrenheit. It could survive in these low temperatures, which is completely bizarre for different diseases in general, because usually they die if it's below 70 degrees, 65 degrees, but here it's going to survive 39 degrees or lower. So it's really important that we get this under control and make sure that the sanitation is there when it comes to botrytis blight. And the spores are dispersed by water impact and by twisting from the stalks upon drying, and it favors high humidity, moderate temperatures, and low light. And you can control this by heating and venting, sanitation, and reducing plant wetness and fungicide sprays. So really that plant wetness, trying to make sure that that last factor, that environment, remember the disease triangle, so the environment's not there. So making sure that that moisture is not maintained on the foliage. So we want to instead of applying the overhead irrigation, we want to bottom feed the irrigation, and we also want to make sure that air circulation is doing good. So we want to have vents. We want to make sure that the foliage is not touching each other to reduce the moisture buildup. All right, fungal leaf diseases, and you can see a couple different photos here, and you're going to see a very healthy plant, and then usually in the center you're going to see those spots, those irregular spots that are going to be occurring, and they might have like a darker circular circle around the perimeter of the circle, and then you're going to see the spores. If you can look at that center in the right picture, the picture on the right, you can see these like black spots, and those are the spores that are there. So that's a really good indicator that this is a leaf spot disease. Fungal leaf spot diseases really likes those wet leaves. So trying to reduce the wet leaves of the foliage, the overhead irrigation, and then making sure that air circulation is there. And another big one, and I don't think I mentioned this yet, but removing those infected leaves from the plants. So you can see that whenever you're scouting, you find that this plant has leaf spot. Removing those leaves properly and making sure that you come back to a node, having those nice pruners, having a nice clean cut, making sure the pruners are sharpened to make sure the plant produces another shoot quickly and heals and calluses that wound well. So we want to make sure that those are taken care of, removed, and then disposed properly. And you just don't want to throw it in a trash can. If you want to throw it in that trash can, you want to make sure that trash can is being autoclaved, being heated to a certain amount for a long period, at least an hour. Or if you want to burn it, that's another possibility. Because if you throw away these plant material that's infected with different leaf spots and root rots and different diseases, and you throw it out, you can potentially be infecting and spreading the inoculation, the inoculum to other plants once you throw it out. So you want to make sure that you don't increase this issue. So we want to dispose of it properly. All right, now we're going to talk about leaf rust, and leaf rust in general are really easy to determine because you see in the bottom photo those spores, the mycelium is orange. It's like that bright orange, so you can detect it very quickly. The diseases are favored by high humidity and moderate to cool temps, spread by wind-blown spores. So we want to make sure that the plants are not touching, the neighbor plants are not touching each other, that we keep down that the fan when we do have it on. We don't have it at such a high intensity. Maybe we bring it down to a medium intensity so we are not blowing those spores anywhere. And then scouting, seeing if we do find these rusts, and then removing the plant material that are infected with rust, and we remove it and then dispose of it properly. It also survives in living plants and plant debris. So that's another one. We got to remove those plant debris out of there and then dispose of it, and then control with sanitation, discarding plants and removing infected foliage, fungicides, and then planting resistant cultivars. And we haven't really touched base on resistant cultivars in general. Resistant cultivars are plants that have been cultivated specifically because they are resistant to different diseases. So when this occurs, say this daylily rust has been very affected by a certain species of daylily, we will find a daylily that has been resistant to daylily rust, and we will plant that instead of the original cultivar that we were using. So it's really neat what technology has done and how we evolved into this, because we can get these different plants, these different plant cultivars that are resistant to so many diseases, so we can reduce the amount of pesticides that are used, which is huge. It's going to save us a lot of money, and it's better for the environment as well. And here are just some other photos of rust that are on Snapdragon and a couple others, but you can see that we have it's raised, it's lifted from the surface of the plant, so it's very fuzzy to touch, and then it's going to be that bright orange coloration that's going to be in the bottom leaf, but on the top leaf you're going to see that discoloration, the yellowing and the spotting, the chlorosis that occurs on the plants, and you're also going to have spots within that. So the bottom of the leaf, that's where you're going to see the actual mycelium in the spores itself. It's going to be very fuzzy in the bright orange, and then the top of the leaf, that's where the plant is losing its chlorophyll, so it's not going to be green, it's going to be yellowish. As you can see in a couple of these photos, it's a very yellow and almost necrotic in spots. Here is a big one, powdery mildew, and you can identify powdery mildew very easily because it has that white fuzzy substance on the top layer of the leaves, so you can identify that pretty quickly. It goes on the foliage and also goes on the fruit and flower as well. So that's a great indicator. And then with powdery mildew, you also want to be looking for sooty mold. Powdery mildew likes that warm, humid, and dry climates, and it sporulates absolutely everywhere, so it doesn't really matter. It can go on the stem, foliage, it could go on the fruit, and then it has localized infection, so you can be very specific. So you have a whole entire tree, and it starts from the lower right area of the tree, and then it works its way throughout the rest. So that's what it means by localized infections. And then we have downy mildew, which is very similar to powdery mildew in appearance. People get confused a lot of times. It still has that white furry substance that's going to be on the foliage, but it's going to be on the bottom leaf, not the top of the leaf. Another one, instead of it liking warm, humid, and dry climates, it likes cool, humid, and wet climates. And then it sporulates on the underside, and it's localized and systemic. So it's localized like the powdery mildew, but in this case, it's also systemic as well. And being systemic, it goes throughout the whole entire plant, which will make it more uniform, not so specific to one certain area and then going throughout the rest. It's going to be very familiar with the whole entire visually. You can see it throughout the whole entire plant. All right, so bacterial leaf spots, and you can see this by having those different, it's very similar to the fungal leaf spots with bacterial leaf spots. It goes throughout the veins more, and you're going to have it more discolored. So you're going to have, you could see like that yellow perimeter, and then you have like the discoloration more in the center, and that's what's going to be occurring, and then it's going to radiate out. But disease is favored by pre-moisture on the plant, so having the plant the foliage being very wet at times, so we can reduce that by making sure that the foliage is not continuously wet, that we bottom feed, and that we do the proper air circulation. Warmer temperatures and high humidity that the disease is favored in, and it's spread through water splashing. And with all bacteria diseases, it's spread by water splashing and plant-to-plant contact as well. So if we can reduce that water splashing, it's going to be very key. Instead of doing the overhead irrigation, we will do the bottom feeding, and that plant-to-plant contact, moving the plants so they're not touching each other, will be very helpful. I know a lot of places, especially when you are bottom feeding, you are putting all the plants on the same exact, say you're putting it on the ground, and then they put all the plants on the ground, and then they open up these valves, and the valves bring in water. And from that point, the whole entire greenhouse floor is covered in water, and then it's uptake by the root systems by capillary action. And from the root systems, they're bringing up that water. The big downside of this, one, it's on the ground, so it's not sanitary. Another reason is whenever this is happening, if we have one plant that has either a bacteria, has a root rot, whatever it is, we have just inoculated all of those plants with the same exact disease. So we want to stay away from that, even though that is a good thing that they're bottom feeding, not overhead irrigation, but we want to really figure out the best solution for the ground feeding. And that is what we do as well. We have separate containers. We have separate trays for each of the plants, and then we bottom feed that way. There's drip irrigation, which is also a pretty good one. It is costly, but there are different factors to go there. But definitely not having all of the plants on one area where the inoculum could spread for bottom feeding is going to be really important. And then bacteria leaf spot also survives in plant debris. So if we are scouting and we find some foliage that looks like it's symptoms of either a bacterial leaf spot or a fungal leaf spot, and we get rid of these debris, or maybe it's just necrotic spots that we get rid of that's not making the plant look that aesthetically appealing. So once we get rid of that, we want to dispose of it, make sure that we're making the right cut so it calluses over nice, and then we will dispose of that plant debris. And then control for this, reduce water splashing, increase sanitation, and then we use copper fungicides to reduce the spread. And then we have virus diseases. And with virus diseases, you guys know that we cannot cure a virus. So that proactive approach, making sure that we don't bring any viruses in, is going to be very important. Different common symptoms, tissue deformation, stunting, leaf necrosis, and that is dead leaves. Mottling. And mottling is a pattern of irregular marks, like spots, streaks, or blotches that come in like different shades and colors, and that's also mosaic. And then the ring spots occur with different viral diseases, and they look like leaf spots, but they have a more uniform circular shape, and the perimeter is yellow. That chlorosis occurs in the perimeter, and then as you go in, it gets more and more necrotic. So it's going from a lighter brown shade from the outside, and then it's going more darker brown to a more black in the dead center, almost as like a frog's eyeball.